Okay, so I just want to start by saying what a great compliment it is to be grouped into the same cohort as John. I will admit that when I first came here, I didn't know anything. I had no idea about any of the stuff that I'm going to talk about today. But other than that, everything else that Bruce said was grossly exaggerated. All right. Okay. So I'm going to break a little bit from this pattern of today of gushing over how awesome Bruce is and how he supported you on your quest to greatness, etc. And I'm going to say that I'm a little bitter. And I was reminded of this. I'm bitter about a lot of things. I'm just going to talk about one of them. I was reminded of this when Bruce mentioned that a disaster was averted when he convinced Anders not to call Freesurfer Beavis. And I want to say that one person's disaster averted is another person's creativity stifled. And so I was reminded of this amazing Freesurfer t-shirt design that I came up with as a new postdoc in the lab, and that Bruce vetoed. And he said that he didn't want a t-shirt that he wouldn't be able to wear in front of his children or his NIH program officer. And I hope that now that your children are older and our grants don't get funded anyway, that maybe we can reconsider. By the way, I have no idea what goes through someone's mind when their new postdoc presents him with that. Pure horror, probably. Okay. All right. So I stole this picture from Bruce's slides. So this is Brodmann areas, the gold standard that in an ideal world, we would like to be able to reconstruct in vivo if our structural imaging was working great. What is the equivalent of that in the white matter? What is it that in an ideal world we would like to be reconstructing in vivo if our diffusion MRI methods were working great? And the answer is we have no clue. Of course, we know that everybody has a cingulum bundle, everybody has a corticospinal tract, etc. But that's more equivalent to knowing that everybody has a temporal lobe and a frontal lobe. What we're missing is a fine-grained and functionally relevant organizational system for the white matter. And I'm going to argue that we are right now in a position where in the next few years, I believe that not only we will have an organizational system of that nature for the white matter, but also the imaging methods to be able to reconstruct it accurately in vivo. And this is due to a confluence of both the right data and data analysis tools, as well as the right anatomical insights that are coming together. And I'm going to talk a little bit about each of those pieces of the puzzle. So the anatomical insights are from work that we've been doing with Suzanne Haber at the University of Rochester, who has spent decades looking at the organization of white matter bundles in macaque monkeys with tracer injections. And so Suzanne and her colleagues have known for a very long time that the large white matter pathways are not monolithic structures. Each of them is more like a highway with smaller side roads merging on and off at different points along the big highway. So we know, for example, that in the case of the ansonate fascicular zone here, there are many smaller axon bundles from the ventral PFC that join the ansonate fasciculus. Some of them follow it all the way to the other end, to the temporal lobe, and some of them follow the ansonate only part of the way and then jump off to join other pathways like the internal capsule, the corpus callosum, the cingulum bundle, etc. And Suzanne's lab has been working with this macaque tracer data showing that in some of these cases there are topographies of the smaller bundles within these big highways. So the question is, can we apply those rules and can we investigate the existence of these rules in the human brain, given that we cannot inject humans with tracers? And I'm going to show an example of that kind of effort to apply and translate those types of organizational rules to the human brain. These images are from Georgia, who was part of her thesis work in visited Suzanne's lab for a few months and spent those months tracing a single tracer injection under the microscope. So going through every single slide under the microscope and tracing the outlines of these axons. Then she put together that information across all the histological slides by co-registering them into a 3D model. After this, she can clean it up into a set of bundles showing all the big bundles that this particular injection from the anterior frontal cortex jumps onto. And from this 3D model, she can then compare directly the tracer injection results with diffusion tractography from scans that we collect on the same brains that Suzanne has injected with tracers on our 47 Tesla scanner. And using this kind of data, Suzanne's group has shown that several of the topographies that she observes in the tracer data can be replicated with diffusion tractography. So this shows an example of such a topography showing 3 injection sites in the dorsal medial PFC, dorsal ACC, and ventromedial PFC, and how, as they enter into the internal capsule, they maintain this strict dorsal to ventral organization, and how this kind of organization can be replicated with diffusion tractography from our scans of the same macaque brains, and also in humans, once Suzanne has placed the seeds in homologous regions, we still see the same green to yellow to red organization inside the internal capsule. Now, the question is, why do we care? Is this simply an anatomical exercise? And the answer is that actually there is relevance in human disease for this kind of work. So using this kind of segmentation of the anterior limb of the internal capsule that Suzanne came up with based on where the fibers in different parts of the internal capsule came from in the cortex, Amelia Versace and Mary Phillips at the University of Pittsburgh tested for differences between patients with bipolar disorder and controls. And they found that the differences were localized specifically in the one segment of the anterior limb of the internal capsule, the light blue one, that carries ventrolateral prefrontal cortex fibers. So this kind of work shows how the implications of this type of anatomical exercise can be relevant to clinical studies, how it will allow us to get more specific results. So if we average the FA over the entire internal capsule, this would have been missed. It allows clearer interpretation of the imaging findings. So if they tell you that there's a difference in FA in the internal capsule, this could mean a lot of different things. But if they tell you that there's a difference in the part of the internal capsule that projects to the ventrolateral prefrontal cortex, that can mean fewer different things. And also this kind of segmentation and subdivision of the big white matter bundles can be relevant for targeting interventions such as deep brain stimulation and capsulotomy. Another thing that we learned from this type of study is that these rules about the organization of the smaller bundles inside the bigger bundles are all described in terms of the relative positions of these bundles to each other. So this shows for pairs of different cortical regions. For example, here the green is area 45 and the red is area 47, and the other ones are different pairs of regions. The centroid of the bundles from those regions inside the internal capsule. And these are different subjects, 35 human subjects from the HCP, MGH-HCP data. And as you can see, when you look across subjects, in some cases, the bundle from area 47 of one subject might overlap with a bundle from area 45 of another subject. So in terms of the absolute coordinates in template space, and this template space mapping was produced by a nonlinear registration, it is not consistent. So we cannot predict purely based on coordinates in some template space. But if we take the pair from each of these subjects and shift it so that the red point is always on the origin, then we can see that the green points move in pretty much the same quadrant, which suggests that there is a clear pattern in the relative position of these bundles. So which one is medial to which one is dorsal to which. And so this suggests that if we get this kind of organizational system for the white matter, it will not be as easy to reconstruct it in subjects just based on coordinates in some template space. But instead, we might want, instead of trying to contort our subjects into a template, we might want to develop methods that instead use the relative positions of bundles to each other and to the surrounding structures. And luckily, over the last few years, we have been developing exactly that kind of method. And I'm going to talk about two examples. One is the atlas-based approach, where we use prior information from a bunch of bundles that have been manually labeled in a set of training subjects to reconstruct automatically the same bundles in other subjects. And this is what TRACULA does. Excuse me. So TRACULA reconstructs the posterior probability of a bunch of different white matter bundles by fitting the shape of the bundle both to the orientations, the diffusion orientations from the diffusion data, and to the surrounding anatomy of the subject. So this anatomical prior term in TRACULA uses exactly this concept of the relative positions between the bundle and its surrounding anatomical structures from the Freesurfer segmentation, rather than using deterministic ROIs to constrain where the track should and shouldn't go. And the benefit of this approach of defining the bundles simply based on which are its anatomical neighbors is that we are not assuming that the shape or the size or the exact coordinates of the bundles are the same across all the subjects between subject populations. We're only assuming that the bundle goes through the same general anatomical neighborhood. So this does not require exact alignment of every subject to a template. It is robust to individual variability, and it is also robust to differences between the subjects that were used to train TRACULA and your study subjects. They don't have to come from the same population or to have been scanned on the same scanner with the same sequences. And as a result, it has been used on a wide range of populations from childhood to old adulthood, as well as a bunch of neurodegenerative and psychiatric disorders. Another benefit of this formulation is that because of the Bayesian framework that we have, rather than a set of deterministic rules based on ad hoc ROIs, we can extend that framework easily to, well, not so easily, but we can extend it to other types of designs like longitudinal data. And this is what the longitudinal stream of TRACULA does. So here, instead of just having a single set of diffusion data and anatomical segmentation, we have multiple time points from each subject. So we reconstruct the tracts jointly, given their likelihood, given the diffusion data from all time points of that subject, as well as their anatomical priors based on the anatomical segmentations from all the time points of that subject. And this is using the within subject T1 template from the longitudinal stream of Freesurfer, which is Martin Reuter's work. And it allows us to reconstruct the subject's pathways jointly, given the data at all the time points at once. This ensures point-to-point correspondence between the time points. So there is no chance that you will get half of the tract in one time point and then another half of it in another time point and then be unable to compare across time points. And it is also unbiased as it treats all the time points the same way. And because of those attributes, we've shown that it improves both reliability and sensitivity compared to just treating each data point, each time point, as a cross-sectional data set and running tractography separately for each time point. So it reduced test-retest error in FA values across 2 sessions of healthy subjects. And also it increased power to detect longitudinal FA changes in Huntington's disease patients with severe and progressive atrophy. So that was the atlas-based approach. And now I'm going to talk about another approach that is purely data-driven. Here we do not have any prior information about what bundles we want to see in the data and what shouldn't be there. We just have the subject's own whole brain tractography by sitting everywhere in the brain, and we want to group the tracts into anatomically meaningful bundles based on how similar they are to each other. So this allows you to perform whole brain exploratory studies, and this is work by Viviana Silas. And conventionally, the streamlines would be grouped together if they are close to each other in space. However, some things are close to each other in space, but actually project to very different regions. So instead, Viviana's work groups streamlines together if they have similar anatomical neighbors. Again, using that information about the anatomical neighborhood rather than describing the streamlines based on their spatial coordinates on some template space. So instead, the streamlines are described by their histograms of anatomical neighbors, so the left neighbors, the right neighbors, etc. And streamlines are grouped into the same cluster if they have similar histograms. So Viviana combined this with this anatomical similarity measure with the normalized Kautz algorithm for hierarchical clustering, which gives you a hierarchical tree structure of smaller bundles within bigger bundles, just like we know that anatomists find. So at the top of the tree, you would have the whole brain data, and then this is split into kind of left hemisphere, right hemisphere, and interhemispheric bundles, and then these are further split into smaller sub-bundles. So the benefits of using this kind of anatomical similarity that as I mentioned earlier, describes streamlines based on their surrounding anatomical structures rather than based on their coordinates in a template space, is that this is more similar to how a human would label different bundles in the brain. So an anatomist would describe something as being a bundle based on a bunch of axons that move through similar parts of the anatomy, rather than based on how close to each other the axons are. So it turns out that without relying on any training data at all, using only this similarity measure and the subject's own anatomical and diffusion data, we get better overlap between these automatically obtained clusters and manually labeled tracts by a human rater when we compare it to the results that we would get by grouping the streamlines based on their distance. And Viviana took that a step further by using those anatomical neighbor histograms to find matching clusters across subjects without having to co-register them into a template space. And we tested this on the pilot lifespan HCP data from Randy Buckner and Leah Somerville, where they collected scans on subjects aged from 9 to 90. And when we find the corresponding clusters across subjects using their anatomical similarity without registration, versus when we have to register all these subjects from all these widely different ages and different brain shapes into the same template to find the matching clusters, we actually get between 20 and 40% improvement in the overlap of the clusters, as the difference between the subjects that we compare in age goes from subjects that have the same age to subjects that are 80 years apart in age. Okay, so how can we do even better? Ultimately, for both of the approaches that I described, both the atlas-based approach with TRACULA and the data-driven approach with AnatomiCuts, we are still limited by the accuracy of conventional exploratory tractography, because that's what provides the training data that goes into TRACULA, as well as the streamlines to be clustered by AnatomiCuts. So if that data is garbage, garbage in, garbage out. So why does tractography still get things wrong? We've gotten these expensive scanners, super awesome sequences, et cetera. Why are we still making errors? So the state-of-the-art acquisition methods that we have improved both SNR and resolution dramatically, which means that we get very accurate estimates of diffusion orientations. However, the spatial resolution is still limited to about 1.5 millimeters, and this means that there will still be often more than one axon population in each voxel. as soon as we have more than one axon population in each voxel, we are going to have multiple configurations of these axon populations that can give rise to the same orientation distribution function. And this means that as soon as a tractography algorithm reaches this voxel, it doesn't know which way to turn. And so it has to make an arbitrary decision, and sometimes that decision is going to be correct, sometimes it's going to be incorrect. And how can we tell if we're getting things right or wrong? Sometimes things are obviously wrong. So in this case, because the algorithm made the wrong arbitrary decision, it jumped from one bundle onto another, from the corticospinal tract onto the corpus callosum, and then onto the corticospinal tract on the other side. But a lot of things that we see in whole brain tractography data is not as easy to determine if they're right or wrong. And also things that we don't see in the data is also not easy to determine if they should or shouldn't be missing. So the way to get to the ground truth in the human brain, without having to inject every single monkey on the planet, is to use ex vivo data. So I'm going to end with an example of deconstructing a brain ex vivo to allow us to reconstruct in vivo data better. So we identified an area where tractography in humans gets things somewhat differently than what we expected based on the macaque data. So in this region, we expect things to split as they come from the frontal cortex into the corpus callosum and the internal capsule. But instead, we see some other stuff. We see more of a hairy mess there. So we found a subject that had previously, a few years back, received a postmortem whole hemi scan in just a regular 3 Tesla scanner. We found that problem spot in that subject. Gene told us where to get that piece of brain from that subject that happened. The brain happened to be, or pieces of it happened to be at the back of the fridge, kind of unwanted. So we put it to good use. And we scanned this block that is cut here, as well as the block under it, in our 9.4 Tesla small bore scanner at much higher resolutions and also much higher SNR because of the higher field, as well as now having the coil elements much closer to the actual tissue area that we're interested in. This shows the data from the top block, and we see that nice split between the corpus callosum and the internal capsule. And, oh no, why is that missing? Well, okay, you'll see it shortly up here. So then we registered the blocks back onto the whole hemi scan, and this shows the data from the 2 blocks. So this is where actually the tissue was cut, and the 2 blocks were scanned separately, but you can't really tell because these are blocks. They're not like histological slices that can get easily distorted or torn. And once I turn on this ROI, you can see how the nice continuity that we get between the blocks that we scanned separately and how we've cleaned up this tough area compared to the whole hemi data. And this shows one of those blocks, the one with a split between the corpus callosum and the internal capsule. And this is just plain diffusion data, but it looks almost like microscopy. And once we get to this resolution, we get to the point where for a lot of voxels, we have only one major axon population. So then we reduce that crossing versus kissing ambiguity quite a bit. But then we will still get to some points where this resolution that we can get on the 9.4 Tesla, which we've gotten now down to 250 microns, is not going to be enough. And also where we will need some measurement that is independent of water diffusion. And for that, we have been working with Hui Wang and the optics group and getting polarization-sensitive optical coherence tomography data that gets direct measurements of axonal orientations. It's based on the birefringence of myelin. So this is data, I'll play that again. with resolution 3.5 micron in plane. And it took 3.5 days to collect the data that you just saw. So that's why we cannot do it on the whole brain. So that's why we need to start with some prior hypothesis based on regions where we don't get the things that we expect from the anatomical studies and hone in on those regions. So to summarize, I talked about anatomical insights from macaque studies and how they provide a roadmap towards an organizational system of the white matter with how the big pathways are subdivided into smaller bundles. I showed an example of how this can be clinically relevant. And I also demonstrated how rules are preserved across individuals and across species, but specifically when you describe them in terms of the relative positions of structures rather than their absolute coordinates in some template space. Then I talked about analysis tools that take advantage exactly of that kind of neighborhood structure. The atlas-based approach with anatomical priors in TRACULA, as well as the data-driven approach with the anatomical similarity measure in AnatomiCuts. Those two can be further combined, so you could do the data-driven approach on high-quality HCP data and then take those clusters and feed them as training data for TRACULA to reconstruct those same bundles and that same hierarchy in lower quality, clinical quality data. Finally, I showed you how we can deconstruct the brain ex vivo to reconstruct it in vivo. And we've been using that kind of data, first of all, to explore those kinds of topographies of the smaller bundles within the big white matter pathways. This can guide the development of better forward models for diffusion and also provide further prior information for analyzing lower res in vivo data. And I want to end by thanking the people who do the actual work over here and my collaborators. Thank you.